Good afternoon to you all, and thank you for coming. When I came here, only Riyaz and myself were there, so he would have made the presentation and I listened. So there are three students who had worked very hard, faced difficult times in ICRISAT or with me, and finally have graduated. So first we congratulate them. And each one of them will be making a brief presentation of what they have done. So we'll have 25 to 27 minutes of presentation and some time for questions hours, but realizing that we don't have much time, some of the questions could be followed with the individual, with the discussion after we have finished the seminars and then we have the tea. So the first presentation by Riyazaddin, Muhammad. Okay, it's on genetic variability for resistance to fruit fly. So over to Riyaz.
Thank you, sir. Good afternoon to all. My research topic is genetic variability for fruit fly resistance and grain characteristics in post-rainy season sorghums. As you all know, sorghum, it is a C4 photosynthetic plant and it belongs to the family Poaceae.
Now another suggestion, you can directly go to your materials and methods, more time on results and discussion. We all know sorghum is an important crop, but declining in importance. So next. Yeah.
So here comes the objectives. Characterize fruit fly resistant lines and improve genotypes for stability of resistance and adaptation to post-rainy season. To understand the mechanisms of resistance and assess genetic diversity of fruit fly resistant lines and improve genotypes with adaptation to post-rainy season. To understand the nature and inheritance of fruit fly resistance and adaptation to the post-rainy season. In objective 1, we have taken around 90 genotypes and evaluated under 2 seasons, in rainy and post-rainy seasons. And of these 90 genotypes, those are performing well, are showing resistance to fruit fly. Around 30 genotypes were selected, and these 30 genotypes were carried forward further for evaluation as well as to know the type of gene inheritance present in these genotypes for fruit fly resistance. So we have recorded the data on fruit fly resistance, morphological and agronomic as well as on panicle traits. Here comes the data for fruit fly resistance traits. Those are fruit fly dead hearts as well as. So this is the data for fruit fly dead hearts percentage. Normally the fruit fly damage is measured in terms of dead hearts. If you compare with that of susceptible genotypes Varna, you see here it is around 98% of dead hearts are seen, whereas in post-rainy season around 60% of dead hearts are seen, whereas the other genotypes they are showing a moderately resistance to fruit fly, they are having less number of fruit fly dead hearts. So this shows that these are the genotypes they are exhibiting resistance to fruit fly in the post-rainy season. Coming to the resistance mechanisms here, in sorghum it is a criteria that if a fruit fly lays eggs on a sorghum plant, then 100% it turns into a dead heart. Whereas you will see here more number of eggs laid on the plants and more number of plants bearing the eggs, but less number of plants with fruit fly dead hearts. This is a type of mechanism known as antibiosis resistant mechanism, where even though the fruit fly lays eggs, the plants does not turn into the dead hearts. Means some sort of mechanism is acting on the fruit fly which does not allow to grow into adult fly again here. In order to identify the association of traits with fruit fly resistance, correlation studies were carried out where leaf glossy score, leaf sheath pigmentation, seedling vigor and trichome density are in association with fruit fly resistance. Coming to identify the direct and indirect effects, path coefficient analysis is carried out. where we have identified that leaf glossy score, leaf sheath pigmentation, and trichomes and adaxial and abaxial leaf surfaces as well as plant height, they are influencing the shoot fly damage. Major contribution, direct major contribution for shoot fly resistance traits. These are the morphological traits contributing for shoot fly resistance. This is glossy, non-glossy. This is resistant. Glossy is the resistant trait, whereas the non-glossy is the susceptible one. This is pigmented leaf sheath pigment and non-pigmented. And here comes the trichomed, that is a resistant check, that is IS 18551, whereas no trichomes are seen in the susceptible check, non-trichomed. So after identifying all these things, the genotypes, those are performing well in the field conditions. 30 genotypes they are selected, and in order to identify how diverse they are, so we have used 38 SSR markers to genotypes these 30 lines, and found that they are genetically diverse, and as well as the biochemical analysis is also carried out. Using 38 SSR markers with a PIC value of around 0.45 is reported, and they are grouped, these 38 SSR markers group the genotypes into 4 different groups, and here it is the susceptible group, that is Swarna 296B, and here it is the resistant group, that is IS 18551 group, and these are the moderately resistant genotypes. This shows that whatever the material we have used in the study, they are genetically diverse in nature. In order to confirm that with that morphological as well as the shoot fly resistance traits, this is principal coordinate analysis was carried out using shoot fly resistant traits as well as with the morphological traits. These traits also placed the genotypes into different groups, showing that they are diverse in nature, where the susceptible genotypes, they are grouped together even if we take the markers as well as the morphological and shoot fly resistant traits. So we have evaluated those genotypes for biochemical components also, which showed that the genotypes, those are exhibiting lower shoot fly damage. They are also having higher carbohydrate percentage, protein and polyphenols and tannin contents when compared to that of the susceptible check Swarna. Here in this case, we do not find drastic change when compared with that of the susceptible check, but certain high amounts of carbohydrates, proteins and polyphenols were recorded in the resistant genotype when compared to that of susceptible one. HPLC analysis was also carried out in order to identify the flavonoid compounds that are responsible for that deter the insect feeding, which showed here I am showing the 2 chromatograms of resistant one and susceptible ones Swarna. Here you see the different peaks. We have coded around 55 different peaks were generated when the HPLC analysis was carried out. There are several common peaks in between the resistant and susceptible one, where we see higher peak areas in the resistant one when compared to that of the susceptible one. This shows that these are the compounds that are responsible for shoot fly resistance. In order to identify the compounds that are flavonoid compounds that are responsible for shoot fly resistance, we have run few standards where we found that here we have the resistant check IS 18551, that is camphorol. It is reporting camphorol and salicylic acid in higher compounds, which is not seen in the susceptible one. Whereas the susceptible genotypes showing the vanillic acid and ferulic acid, that does not reported in the resistant genotype. This shows that these are the compounds responsible for susceptibility, and these are the compounds responsible for the resistant nature in the genotypes. So even though few of the genotypes that has reported these compounds, the concentration varies within the genotypes. Coming to the objective 3 here, to understand the nature and inheritance of shoot fly resistance with adaptation to post-rainy season. Based on the molecular diversity, biochemical diversity, as well as morphological and agronomic diversity, we have selected around 10 genotypes and crossed in a full diallele manner, and we have generated around 90 F1s and evaluated along with 10 parents under two field seasons. So the same data on shoot fly resistant traits and morphological and agronomic traits are recorded. And coming to here, These are the genotypes: 700 Phule Anuradha, 25019, 2123, 2146, and these are the genotypes with high glossy vigor, leaf sheath pigmentation, and high trichome density. And these are the marker traits that were reported in the previous slides for shoot fly resistance. These genotypes, they possess all those traits, whereas these are the crosses. They are having a high leaf glossiness, leaf sheath pigmentation, vigor, and high trichome density in the post-rainy season. And these are the genotypes. These are the common genotypes that are seen in the crosses where the resistance is reported in the crosses. And here these are the genotypes. They are having the antibiosis resistance. That is, even though they are having the shoot fly eggs, but they are not turning into the dead hearts. That is antibiosis resistance. These are the genotypes having the antibiosis resistance mechanism in the post-rainy season. Coming to the mean squares of general combining ability and specific combining ability studies here, the mean squares, the significant mean squares were reported for most of the traits for general combining ability and for specific combining ability, indicating that both additive and non-additive gene interactions are involved in the inheritance of these traits. Few of the traits, they are having significant reciprocal combining abilities, indicating that maternal inheritance is involved in the inheritance of these traits. Coming to the diallele analysis of this agronomic and panicle traits, here also the same criteria, same thing, since significant GCA and SCA combining abilities are there, both additive and non-additive gene interactions are involved, and for days to 50% flowering across seasons, and plant height, 100 seed weight, and panicle compactness, shape, and bloom color, they are having the significant reciprocal combining abilities, indicating that maternal cytoplasmic influence in the inheritance of these traits. Coming to the GCA effects here, you see the similar pattern for CSV15, which is showing the susceptible nature, with that of the Swarna. It is also a susceptible genotype. They are having the similar pattern, whereas the other genotypes those are showing moderately resistance or resistance to shoot fly. They are having the negatives and significant GCA effects, whereas these are the shoot fly traits, and these are the marker traits that are responsible for resistance. So most of the genotypes, they are having significant and negative GCA effects, indicating that these genotypes can be effectively used in developing the shoot fly resistant sorghums. Coming to the agronomic and panicle traits here, for days to 50% flowering and plant height, these Phule Anuradha, CSV15, 25019, PS35805, even Swarna, in order to decrease the height, plant height, and decrease the maturity period, we can use these genotypes for developing the shoot fly resistant sorghum with lower maturity and lower plant height. For 100 seed weight, these are the genotypes. Those are increasing the with the significant positive GC effects. These increase the 100 seed weight, as well as these increase the grain yield. Hence, these genotypes can be effectively utilized in increasing the yield traits in the sorghum genotypes. The other panicle traits which are having the positive or negative significant GCA effects can be effectively used either increasing or decreasing of the traits, expression of the traits. Coming to the genetic parameters for shoot fly resistant and morphological, that is variance components. Here the shoot fly resistant shoot fly traits, they show higher SCA variance and higher dominance variance and lower predictability ratio. This shows that predominance of a dominant gene action is involved in the inheritance of the shoot fly resistance traits. When coming to the marker traits, that is glossy score, plant vigor score, they are having the higher predictability and higher GCA by SCA ratio with lower SCA variance and dominant variance, indicating that predominance of additive gene action involved in the inheritance of these traits. Coming to the agronomic and panicle traits here, these days to 50% flowering, plant height, and 100 seed weight and grain yield, they show higher SCA variance, whereas the GCA by SCA ratio and the predictability ratio are high for almost most of these traits, indicating both additive and non-additive inheritance pattern. Whereas for grain yield, the significant SEA variants and significant dominant variants indicate that predominant of dominance gene action is involved in the inheritance of grain yield traits. So coming to the conclusions, the genotypes exhibiting resistance to shoot fly in the post-rainy season as well as across seasons can be effectively used in breeding the shoot fly resistant sorghums with adaptation to post-rainy season. Expression of resistance to sorghum shoot fly exhibited season specificity and utmost care should be taken while selecting the parental lines. Now, based on the molecular diversity, whatever the genotypes that are showing highly diverse in nature, they can be utilized in the breeding program for shoot fly resistance. In knowledge on increase and decrease in the levels of biochemical components can be useful to select the shoot fly resistant sorghum. The marker traits that are leaf glossiness, leaf sheath pigmentation and trichome density can be used for selecting the shoot fly resistant sorghums. So the genotypes that have performed well even in the process as well as individually can be effectively used in developing the shoot fly resistant post-rainy season sorghums. Both we have reported both additive and non-additive type of gene action controlling the expression of the traits. So predominance of dominant gene action for shoot fly traits indicate heterosis breeding is ideal for improving the shoot fly resistance in sorghum. The predominance of additive nature of gene action for leaf glossy score, those are the marker traits. Suggested that recombination breeding with pedigree method can be used for incorporating these traits in sorghum cultivars for improving the post-rainy season with shoot fly resistance. So these are the publications, one accepted in plant breeding and remaining are submitted in international journals. These are the seminars. So I thank all Sorghum Entomology Group for their support during my PhD period. And
the breeding as well.
Yes. And Sorghum Breeding Group. And last, Bill and Melinda Gates Foundation, which has supported for my doctoral study. And also LSU as well as Housing and Food Section and my professor from Usmania University, Dr. Kavi Kishore, and all my professors from Usmania University. Thank you.
So, thank you, Riyaz, for doing an excellent job. Not only good work, but also presenting in allotted time, 20 minutes. So now we have 10 minutes for questions.
So among the three traits you mentioned, morphological traits, so if you have to pick one, I mean, which one has more importance as compared to others? Another question is, all these three go together or they are different in different lines?
Morphological traits.
Yeah.
They are different in different lines. That is what I have shown, that GCA effects and SCA effects. If you are having the predominance of dominance inheritance, you are having the additive inheritance. So again, if you plan for a breeding for these traits, there should be some specific strategy, and proper line selection should be there.
So in the metabolomics, there was a peak around 35, which was in the resistant line only. Do you know what's that metabolite particularly?
No.
It's unknown?
Unknown compound.
Okay. Yeah, well, we have some idea of these compounds, but we still need to know the many others. So there is another paper by Shiv Kumar Chamarthe in Journal of Biochemistry and Molecular Journal of Biochemistry and Biotechnology, Springer Plus. Okay. That has more information of the compounds that are involved. Questions now, students. Shiv Kumar has to ask a question. Yep. All the students. Those who don't ask, they will fail. Come on. No, seniors are not allowed. Go ahead.
Excellent. Let me congratulate you. Very in-depth study, and you have covered a lot of areas. You also mentioned one of your conclusions: seasonal variability and resistance. Again, this key insect on sorghum is really, its population dynamics also very season to season, even the season monthly variation. So how your findings are really useful to our Telangana farmers who are eating 90% of the families, in 80s, sorghum became endangered crop in this region. So how this is going to really bring up back our sorghum in Telangana area?
It all depends on how we select these, whatever the study we have carried out and how the traits are influencing the shoot fly resistance and how we procure all these things into the hybrid. Since inheritance is individually dependent on the traits, shoot fly resistance, they are going for dominance, whereas the others, they are having the additive influence. So if we can plan a strategy where we can bring all these things into a hybrid, I think we can cope up with that.
There were more number of questions than I could have answered in 1 hour. In 10 minutes, they asked so many questions. So those young children are much more attentive, thinking, and speculative. I presume you still retain some of that enthusiasm since you want to be scientists.
Maybe Riyaz has not planned that one.
ये इन सब का एक दिन का scholarship or salary will be deducted, and you will pay that to me. Okay? Those who have not asked a question.
How much you are compromising on the taste and yield and quality, Dr. Sharma mentioned. Those are the key things.
We haven't gone for taste, but yield we are working on. There are certain crosses which are having high yield, I think, and also showing resistance to shoot fly. I think we can select those lines and we can forward.
Okay, let me add one thing. Riyaz has done a tremendous amount of work. Each of his trials on inheritance will occupy nearly 1 hectare of land, and he collected data on about 45 traits, 50?
35.
35 traits. So he was busy from morning till evening, 365 days, 24/7. So that should make him an excellent breeder. Thank you. Let us
Thank you.
applaud him.